So good morning to everybody. Thanks to the organizers and for the chairs for having me here. I want to present a novel autoantibody today to you, which is an autoantibody against the major Volt protein in systemic lupus. And in the slides I will lead you through our discovery and verification validation efforts. In this session I don't need to go too much into SLE being an enigma and a beast with lots of different factors contributing to autoantibodies in vast amounts and tissue damage in almost any tissue you can think of. Difficult to diagnose, variable clinical course, different responses and poor responses to different treatments. Diagnosis of SLE is very much supported by looking at autoantibodies. We all know that anti-dsDNA is a specific diagnostic marker for lupus, but only occurring, depending on the test you use and on the longevity of disease, in 40 to 70% of patients. Others occur in lupus, but also in other diseases, so there's a mishmash of some specific markers which do not really lead to 100% clear diagnosis, and supportive antibodies which have their appearance also in other diseases. So we think that it's clear that it's a multifactorial disease, very complex with multiple organ effects, variable clinical presentation, remitting relapsing disease, only one drug approved over the last decades. Still many pharma companies are active in developing drugs against SLE, which is a very good sign, but they need support by better diagnostics, better biomarkers. So we set up our zero-tag screening technology, which was reported in other presentations at this conference, where we have up to 8,000 human protein antigens. We code them to magnetic beads, Luminex beads. We can mix up to 500 different protein-coated beads and then incubate patient sera. We typically do that in study sizes of hundreds of patients. Here in that SLE study, we used about 7,000 human antigens, 700 SLE, 1,000 healthy controls, and about 500 other autoimmune diseases. Did further replication verification studies to then appear with a set of novel autoantigens. In specifics, in the discovery study, 130 SLE, more than 100 significant markers at a certain specificity level. In a verification study, 183 SLEs, another validation study, 100 SLE patients. In the end, we could come out in those different cohorts with a number of 47 novel autoantibodies, adding to the already 100 or 180 that are in literature. When we look for pathway level relationships of the novel autoantibodies, we see that they spread over many different Go terms. The response to type 1 interferon is one of the hallmarks in SLE. Response to interferon gamma, but we also have innate immune response, humoral immune response, chromatin assembly, showing that just the diversity of the disease. This plot gives you a selection of the top 20 autoantibodies found. Naturally, we did include double-stranded DNA, SSA, anti-P, SM, and so on, also serving as benchmarks, and later on serving as comparators, whether those on the right-hand side, which are a few of the novel ones, tell different stories or are just me too antibodies compared to the known ones. In here you see MVP, major Volt protein. In the first study it had a prevalence of 20%, in the next one 30%, in the third one about 15% prevalence. So there's a difference between cohorts, but that also is in inclusion criteria. You may note that in double-stranded DNA there's only 2 bars because in one study inclusion criteria it has to be double-stranded DNA positive, so we did not find it meaningful to put 100% on here. The major Volt protein itself belongs to the largest protein complex in the cell. It's the 13 megadalton Volt complex. It looks like that. It has a hollow barrel structure, so in the inside it looks like a cathedral you may think of. Therefore it's called the major Volt complex. 96 copies of MVP plus other minor Volt proteins and small Volt RNAs are in this protein complex. It's highly expressed in macrophages. Its function, it's involved in multi-drug resistance, nucleocytoplasmatic transport, cell signaling, and immune processes. It's induced by viral infections, which we find is quite interesting. It's an interferon gamma responsive gene, and its cellular localization is to be seen in the protein atlas, where the green dots show it's a cytoplasmic localization. We haven't done indirect immunofluorescence yet in order to find out what does it look like in IEF. Now let's look at some data. This is from a cohort where we had about 90 controls, 40 Sjögren, 50 systemic sclerosis, and 108 SLE patients. You see two ROC curves. One is the protein as we use it in our discovery screens. The other one is after some protein optimization, further purification of the antigen. And we see that from 19 to 26% we could increase the sensitivity of that protein at a specificity of 0.97. If we look into different diseases, we then see, okay, in Sjögren, in healthy control, in systemic sclerosis, there's spurious positive ones, and clearly the majority with very nice p-values shows that it's a specific SLE antigen. And if we go and compare this antigen and its reactivity to other known antigens like double-stranded DNA, anti-SMD, or anti-ribosomal, we see certain areas of overlap, but we also have, out of 27 positives, 7 are just triple negative in comparison to the others. So we believe that this is about 10% of SLE patients will have that autoantibody and are free of those other hallmarks. So we think there's a realistic chance that the sensitivity of lupus diagnosis can be improved by MVP. And I have also to express my hope that some of the other 46, we have not been as far advanced as with this one right now, will help to further close the gap. In a presentation yesterday, I have shown that this collection of autoantibodies is capable of segregating SLE in subgroups. We believe that MVP is a strong factor contributing to this subgroup segregation of SLE. So my summary: high content autoantibody screening reveals multiple novel antigen targets. We have done that in SLE, we have done that in Sjögren, systemic sclerosis, rheumatoid arthritis. I'm looking for further indicational fields. MVP seems to be a specific SLE antigen with prevalences between 17 and 30%, depending on the cohort analyzed. We have not looked in other ethnicities outside of some African Americans, some Hispanics, and the majority Caucasian. So we would certainly be interested to look in the Southern Americas or in Asian populations. So if one of you is interested, please give me a note. We see that patients negative to the hallmark antigens have reactivities against MVP, so it seems there's an additive value. And certainly we need more studies, and collaborations are welcome, and one of our missions here is to find people interested working with us. Our technology, Luminex-based, is transportable, so if somebody has a Luminex machine, we can provide protein-coated beads. Also, it is a very hungry instrument. We can analyze up to 2,000 samples per week, so even big longitudinal studies are available to be analyzed on our system. With that, I want to conclude and thank especially our main collaborator, Professor Matthias Schneider. He is the head rheumatologist in Düsseldorf, and he has opened up his resources and made many connections to other key opinion leaders to promote our work. And with that, I thank you for your attention. I'd be happy to take any questions.